It contains all of the chemical digestion needed to break down that food product. The fourth area is called the pyloric region. It's the most inferior aspect and that area in which the chyme will exit out of the stomach. It also contains that pyloric sphincter that regulates the passage of chyme. And again, it's a bolus coming in, it's chyme when it leaves. There are the parts and pieces. There's the cardia, there's the fundus trapping. The clinical term for burping is actually belch or belching. So make sure you know the clinical term because you will have to write it up in histories for your patient. There is the body and there is the pylorus, okay, leading directly into that duodenum. I do want to show you this. Look at the layers running in, and these guys almost 100, well, these guys definitely 180 degrees to each other. And I don't necessarily care that you know the names of the longitudinal, the circular, and the oblique muscles. I do, however, need you to know the function of them. When I have 3 different muscle directions coming together and contracting all at the same time, what happens is that bolus that's inside that stomach gets contracted and rotated in 3 different directions, providing for us 360 degrees of rotation to be able to break down that food material. Inside the stomach, you will find what are called rugae, and the rugae are the really textured area that are created by the cell types that are found inside the stomach. We'll talk about those in just a minute. This is the greater curvature, lesser curvature, the rugae that are found inside, fundic region, cardiac region, 3 types of muscle, so make sure you know those. Here's what the actual cells inside the stomach look like. Do you see all these hills and valleys? Now, these are not villi like you'll find in the jejunum and ileum. These are called gastric pits. And the gastric pits, I'm going to show you in just a minute, contain 4 types of cells emitting chemicals that help us break down and digest the material that's there. Okay, so those gastric pits, simple columnar epithelium. This is what they look like. There's a pit, 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 pit. What we find in the pits is what's most important. So here we go. Number 1, the mucus cells. The mucus cells secrete mucus. You may also hear them referred to as goblet cells. Okay, so right there they are. Mucus neck cells or goblet cells. Large amounts of mucus to lubricate and to create texture in that chyme that's being broken down. Number 2, what we call stem cells. Stem cells found down in the bottom or in the base rapidly divide to provide us with new cells on a regular basis. The parietal cells, these little brown ones right here, and this is what's really important, hydrochloric acid. Also, intrinsic factor. So they emit hydrochloric acid and what's called intrinsic factor. And we'll talk about intrinsic factor later in the semester. So those are the parietals. The chief cells. The chief cells are these little pink guys right here. And they're creating something called pepsinogen, which will be converted into a product called pepsin that helps us break down our protein-based materials. Okay, so chief cells. Pepsinogen converted to pepsin that breaks down protein-based materials. And then the last of these is called an enteroendocrine cell. I'm not as concerned about that, although you do see the example of the paracrine messengers to help regulate digestion and to tell you how full you are. Okay, there's a look at all of them. Let's talk about the hydrochloric acid. And really what I want you to know, now it goes through this whole conversion of ferric irons to ferrous irons. What I want you to know, 2 things. Number 1, it will break down pathogens. So it's a protective mechanism by our immune system to eliminate any unwanted microorganisms. Number 2, pepsin and pepsinogen help to break down protein. Okay, so don't worry about all this and the way that it's converted. This right here, pepsin digests protein, and that's really important. Okay, gastric lipase we talked about a little bit. 10 to 15% of the dietary fats are broken down in your stomach. Not much. The bulk of lipids are broken down in the duodenum. Intrinsic factor, just real quickly. Intrinsic factor is essential for B12 and the absorption of B12. And without it, you cannot absorb up B12. B12 is really, really important because, and it says, needed to synthesize hemoglobin. Yeah, to mature out red blood cells, we must have B12. we talked about ulcerations, and we talked about Helicobacter pylori, and the clinical condition that it can occur because of a microorganism that gets trapped into the gastric pits. The researcher, the developer, who pinpointed Helicobacter pylori, actually wanted to run a test on patients, infect them, and then go through the process of killing off that infection. And the FDA said, no, you can't willingly and knowingly infect people as an experiment. And so he agreed and moved on with life, but he infected himself. And so this is the first time that we've seen a scientist who really believed enough in his work to infect himself. It's not the first time, but it's the most recent. This research was done maybe 15, 20 years ago. He actually infected himself. He ended up with the symptoms of Helicobacter pylori, and then went through the process to heal himself through the pump inhibitors and the administration of antibiotics, and worked very, very, very well. Okay, so what we call peptic ulcers or ulcerations are caused by Helicobacter pylori. It is a microorganism. It is a bacteria that you will find, and again, treated with antibiotics and pump inhibitors. So let's move on and talk about some of the other characteristic features. That guy right there, the liver, and underneath it, the gallbladder. So we'll talk about the liver and its functions. The liver is split up into 4 different pieces. There's the right lobe, it's large. There's the left lobe, it's kind of medium inside. What they call the quadrate and the caudate nucleus right here. Now, I'm not going to necessarily make you memorize the quadrate lobe and the caudate lobe, but I do want to show you this guy right here. That is the gallbladder sitting on the posterior inferior aspect of the liver. This is what the individual unit, functional unit for the liver looks like. So you're looking at a hepatocyte. And the hepatocyte is very, very, very infiltrated with blood, as well as lymphatic materials and arterial materials. Remember, the liver is the largest blood filtration point in the body. Yes, the spleen also is notable. However, it's not the biggest of the filtration devices that are there. Okay. The other thing I want to point out is that with the liver and with hepatocytes, you are looking at sinusoidal capillary beds. And again, the sinusoidals are the ones that had really, really, really big holes in them. And we talked about the fact that they are on-ramps and off-ramps for the blood that's being delivered into that area. Okay, so there are the hepatocytes releasing or absorbing glucose, amino acids, iron, vitamins, metabolism and storage. They also break down the stored glycogen. They are a huge filtration point, so degraded hormones, toxins, bile pigments, drugs. And then it also will release huge numbers of proteins that are unbelievably important for the maintenance of your body. Number one, albumin.